All right, hey everyone, welcome to the final exam review session. I just sent not long ago the Micro 1 review PowerPoint that we're going to use while we go through this. So you'll find that in your email. But basically we'll just kind of jump into it. So just a quick review on a couple terms that we first learned way back in like week 1 and 2, and hopefully you guys have been using these easily the entire term, so none of these definitions should be new to you. But just to quickly review, again, if you're listening to this as a recording, just pause if you need to type stuff out. I'm not going to type out with these, I'm just going to kind of verbally define them because I feel like you should know these by now. But if you need to pause while we're going through, that's the beauty of listening to a recording. All right, so normal flora, again, are just those bacteria that are normally found in or on the body that are part of the immune system that are good for helping protect our body, I guess. You know, they're good bacteria for us. Colonization is always defined as the persistent survival of microbes. Opportunist is any bacteria that sees a situation where they can flare up and take advantage and really multiply even more and then cause disease. Virulence, that is how disease-causing a bacteria can be. When we say the more virulent a bacteria is, the better it is at causing disease. Typically, it has all those virulence factors that go with it that really help it to cause disease. So we've learned those with various bacteria, like they might have a capsule or they might produce toxins and be able to have the ability to evade phagocytosis, that kind of stuff. Pathogen just means that it does cause disease. That's always referred to as disease-causing. Nosocomial is hospital-acquired. So these are hospital-acquired infections. And then fastidious, which we just talked about, some fastidious bacteria at the end. Those, again, are bacteria that need extra stuff to grow. They need extra nutrients. What we always say, they're more picky. All right. So some stains, you of course have been gram staining all quarter, so this should be very easy for you to get. Remember, there are 4 reagents within the gram stain. There are crystal violet, iodine, decolorizer, and then safranin. So those are the 4 reagents. At what step can you differentiate a gram positive from a gram negative? And again, that would be the decolorizer step. And what makes a gram positive different than a gram negative is the peptidoglycan layer within the cell wall. Again, gram positive have a thicker layer of that peptidoglycan, and it also contains teichoic acid crosslinks. Those things will really embed that primary crystal violet stain. So when you go to decolorize it, it doesn't rinse off. It stays purple. That's why gram positives are purple. Whereas gram negatives just have a thinner layered peptidoglycan, so when you decolorize, it easily rinses that violet stain off. And then when you counter stain it with the safranin, that pink color, that is the stain it retains. So therefore, gram negatives are always a pink or red in color. Okay, acid-fast stain, we did learn this back in the beginning. We haven't talked a ton about acid-fast. We did have one chapter a little bit on it, but next quarter we'll have mycobacteria, which are acid-fast positive. But in general, you should just know that there are 3 reagents within acid-fast stain. It's carbofuchsin is the primary reagent, which is again a bright red fuchsia color. That is the positive for acid-fast. Then it's an acid decolorizer, and then it's the counter stain methylene blue. So again, if something is considered acid-fast positive, they will keep that carbofuchsin bright pink or red color to be positive. If they are considered acid-fast negative, they will keep that counterstain blue color. There's 2 methods to acid-fast staining. There's the Ziehl-Neelsen and the Kenyon methods. The primary difference between those 2 methods is, again, Ziehl-Neelsen uses heat as parts of the acid-fast, whereas Kenyon is known as the Coles method because it took away the heat requirement and instead used increased amounts of phenol to do what it needed to do. And the reason that we acid-fast stain is that there are certain bacteria out there, like the mycobacteria whole group that we'll learn next quarter, that have a different cell wall makeup than normal. And in their cell wall, they have mycolic acid. So again, acid-fast stain is specifically targeting that mycolic acid in the cell wall. they won't gram stain properly because, again, the cell wall is made differently. And then finally, we had some fluorescent stains that we covered. You just have to simply know what these each target. Auramine, rhodamine will bind to mycolic acid. So again, anything that would be acid-fast positive would also stain positive with auramine, rhodamine. So both acid-fast stain and auramine, rhodamine are used for the same purposes. Acridine orange is a fluorescent technique in which it will specifically bind to nucleic acids. This is a good one for that last chapter where we just learned mycoplasma and ureaplasma. Remember, those are unique because they have no cell wall, so they're not going to gram stain at all. If you have no cell wall, you can't gram stain it. But they do have nucleic acids in them, so they could use acridine orange to really help bind to the nucleic acids and detect that they are there. Chalcofluor white is binding to the walls of fungus. All right, moving on. Next, we learned all about our augers. Again, there are 4 auger categories that you must be familiar with. Supportive just means it will support the growth of almost everything. So again, almost anything will grow on augers that are considered supportive. The items that won't grow would be the fastidious ones, the ones that are extra picky, and that's where enrichment comes in. Enrichment is just that. It has been additional nutrients, additional stuff put into the auger to really help those fastidious, picky organisms thrive on that auger. Selective has different antibiotics or other chemicals, whatever, added to it so that you specifically grow just one category of organisms. Common examples are maybe it only grows gram-positives or the auger only grows gram-negatives. And then differential is it kind of subcategorizes the bacteria that's growing into different, types depending on their characteristics. So one characteristic that we use a lot with differential is fermentation. Some that ferment versus those that do not ferment. So they'll produce different colors to really help them become subcategorized. So just to kind of learn more about what's growing. All right, so let's go through our different augers that we've learned. Remember what categories we put in there and anything extra that we need to know about them. I'm just going to do all the talking through this just because I don't want to force anybody to be put on the spot that's in this right now. Again, I will type out as we go, but you can pause it to catch up as you need. So we're going to, of course, start with our most common auger used, which is the blood auger. You might also see the initials SBA for sheep blood auger, same deal. Again, it fits into 2 categories. It should definitely be supportive because we use blood auger the most as it does pretty much grow everything. As far as the other category, it would be considered differential, and that is because of the hemolysis patterns that certain bacteria can take up, so it's that subcategories. So remember, with hemolysis, you have beta, which is the complete clearing around the organism, so it has that halo effect, which hopefully you've all seen that now in the lab. Alpha, which is that partial hemolysis where it's more green in color, and then gamma, which is basically no hemolysis at all. I don't know why, I'm just going to get rid of that. Okay, anyway, I'm going to stop playing with it. All right, chocolate auger is also supportive, grows pretty much everything, and actually it's enrichment. So it really will grow everything, including fastidious. I can't spell clearly today. And that's all we need to say there. Thioglycolate broth, maybe some of you guys have seen this now in the lab. Not all labs will play with this, but if maybe one of your labs had it. It's a liquid auger in a glass tube instead of being on a plate. And this is supportive and enrichment as well. So pretty much is just a really good extra media to use as a backup. Some things just really love to thrive in there. So a lot of times we'll plate this out like with different auger plates just to have as a good extra source. So everything. And this is the one that sometimes, now it's not always textbook perfect, but sometimes has different growth patterns in that liquid depending on what grows. So remember, sometimes you might see little like kind of fuzzy, cloudy little balls growing when it has gram-positive in it. Sometimes it can be cloudy throughout. That's usually just an indicator that it's bacteria growing, but they say it's diffuse or cloudy throughout, especially when it's gram-negatives, but I kind of think any bacteria can cause that, gram-pos or neg. If it's only cloudy at the top, that is saying it's a strict aerobe. It has to have oxygen, so it wants to get all the way up to the surface to be next to that oxygen. And then the opposite, if it's only cloudy at the bottom, that's indicating that perhaps it's an anaerobe trying to get away from the oxygen. All right, CNA, I think you guys have all used CNA or PEA in your labs this quarter. CNA is, again, selective, and PEA is selective. So we typically only have one of these on hand as they do the same thing. Both are going to grow gram-positives. And that's all we have to really say there. Okay, so that's the first slide done. Hopefully that was a little simpler for you guys. Those pretty much should be familiar. Going to the next one, MacConkey agar. I know we definitely have all looked at this at some point. These are selective and differential. And it is selective because it's growing gram-negatives, typically gram-neg rods to be specific. So that's the selective part. The differential part is that it will separate the growth based on lactose fermenters, which tend to be purple in color, and non-lactose fermenters, which tend to grow as a clear or colorless colony. HE agar stands for hectoenteric. Again, same categories, selective and differential. This is looking for Shigella and Salmonella specifically, which are both gram-negatives. So that's the selective part. It's looking for just the enteric gram-negatives, which means the intestinal kind of growth, but specifically Shigella, Salmonella. And then again, it's differential based on fermentation. Now, when you do have Shigella, Salmonella growing on this plate, they will appear blue-green in color out in the colonies. They are both non-fermenters. And then an extra little note, remember Salmonella is huge with producing H2S, which will cause the Salmonella to have a black precipitate. So it might have like a black center in the colony on it to represent that. And again, XLDs can be the same purpose. So most labs are just going to have one or the other. So selective differential grows against Shigella and Salmonella is what we're looking for. So again, the differential part is based on fermentation. So on these ones, I believe the Shigella and Salmonella will be clear colonies. And again, the Salmonella will have that black center. Syn agar is a selective agar that is specifically looking for Yersinia enterocolytica especially, but you could just say Yersinia. And this is the one where they have the bullseye appearance to the colony, like that target symbol. TCBS is selective, and this is specifically looking to grow Vibrio, especially Vibrio cholera, which is the big pathogen of Vibrio. I know we haven't really talked about these last couple organisms, but you should still be aware of the agar. Okay, continuing on. These next two you definitely should be aware of. Thayer-Martin and Martin-Lewis are both selective. And enrichment, which is kind of a unique little combo. And they are specifically selective because they are both looking for Neisseria. And that should make sense if you remember that Neisseria is a very fastidious little guy, so that's why they're also enriched. They got to give those extra nutrients to make sure that Neisseria wants to grow. Now, they have all sorts of antibiotics in them to make sure that nothing else really grows except the Neisseria. So again, you got to know these. So it has colistin in each one. Again, that inhibits gram-negatives from growing. They have vancomycin in each one, and that inhibits gram-positives from growing. And remember, the Martin-Lewis actually has more vancomycin than the Thayer-Martin. They both have trimethoprim to inhibit Proteus from taking over and really swarming the slide. Trimethoprim, Proteus. All right, and here's where they really differ. In the Thayer-Martin, it has nystatin to inhibit yeast from growing. whereas, again, in Martin Lewis, it has mycin to inhibit the yeast. So make sure you are still familiar with those. For Day Ginkgo, you guys have answered a question on this on your study guides. We've talked about this. Again, it is a selective agar specifically looking to grow Bordetella, especially Bordetella pertussis, to detect whooping cough. There are other ones that will grow Bordetella as well that we've discussed called Reagan Low. And then Jones-Kendrick is more of, I think, a transport kind of agar, but you can talk about it anyway there. All right, Lowenstein-Jennings. We really haven't mentioned a lot about this at all. The only thing I'm going to say is it is selective. We will learn it more next quarter, but this is really there to grow mycobacteria. So again, we'll get more into that next quarter. BCYE is selective and enrichment because it is growing the little fastidious guy, Legionella. So again, Legionella needs L-cysteine and iron. That's why it needs the enrichment part to grow. And we had that whole discussion post on Legionella, so hopefully Legionella is a little bit more familiar to you guys now. Okay, so that's plenty of agars. There, of course, is a lot more out there that we haven't really discussed yet or that we haven't brought up here yet, but as we go, if I remember any extras, I'll make sure to point them out. All right, next we got into our first set of bacteria, which were the Gram-Pos cocci, and so basically staph, strep, and enterococci were our buggers that we played with all quarter. So after we knew it was a Gram-Pos cocci, we always ran the exact same test every time, which was the test to separate between staph from strep, and that was the catalase test. Again, staph should very much be catalase positive, and all streps are catalase negative. This should be easy for you right now. And you guys should, of course, remember that the reagent you used was hydrogen peroxide, which is H2O2. Okay, so that should be easy answers. Here's some of the staphs that I want you to know for the final. Staph aureus, of course, since it's our big bad pathogen of the bunch. This is very much a medium to large colony. Again, staphs tend to be a little bit larger colonies than streps. They're a little creamier looking. Staph aureus tends to sometimes have a yellow hue or yellow tint to the color. Now, you may have seen that, you may have not. It's not always picture perfect again, but that's what aureus stands for, is gold, that yellow hue. And it is very much beta hemolytic typically. All right, again, catalase positive. The minute we know it's catalase positive, we should be performing a coagulase test next. And Staph aureus is hugely coagulase positive. Remember, there are two types of coagulase testing that you can run. There is the slide coagulase testing, looking for the bound coagulase enzyme, and it'll appear as agglutination or clumps. And then there's the free coagulase, or tube, I should say, tube coagulase test, looking for the free coagulase enzyme, and that'll look like a little fibrin and gelatin-like clot. So again, not every lab will run two. Those that do run two usually use one as a confirmatory method for the other. Again, disease of Staph aureus, pretty much anything. It's a huge pathogen. So you have lots of different skin diseases, wound infections, UTIs, toxic shock syndrome. I mean, the list just goes on and on and on and on. So we're not going to get too much there. Remember, there is MRSA, though, methicillin-resistant Staph aureus. So this is Staph aureus that has become resistant to the psyllins, like the beta-lactams. And so typically we will treat with vancomycin in that case for MRSA. Extra, let's see, Staph aureus is found as normal flora in the nares, which are like your nostrils. A little skin, not a lot. I don't want you to remember a lot there, that kind of stuff. Okay, I think I feel good on that right now. I might come back and add something else here in a minute. All right, Staph epidermidis, or Staph epi for short. You guys, I think, have all seen this already this quarter, so you should know it's a little bit medium. They tend to be white or gray colonies that are non-hemolytic. Again, catalase positive, it is a staph, so it's definitely catalase positive. When we coagulase test it, it should very much be coagulase negative As far as disease goes, a lot of times this is contamination because it is such a huge normal flora on the skin. That's why it's named epidermidis, for the skin. But if we do need to link it with the disease, when it does tend to cause disease is with those prosthetic or medical devices. Remember, Staph epi has the capability of producing a slime as part of its virulence factors. So when it does that, it does this whole thing that we call biofilm, where it can really hang on. And if we don't have those medical equipment really cleansed really, really well, like sterile, and that slime layer has hung on to there and it gets introduced into a sterile area of the body, then it allows that Staph epi to really thrive and make an infection at that time. Okay, Staph sapro basically looks just like Staph epi, white-gray, non-hemolytic. So they look alike. They're going to start testing alike, catalase positive, but coagulase negative. But in this case, Staph sapro, as I'm sure you guys all remember, this one always sticks out with people, is it very much causes UTIs, especially in younger women. And that's because it is found as normal flora in the genitourinary tract, and upon sexual activity, it can get introduced further into that urinary tract to cause a UTI. So because Staph epi and Staph sapro tend to look a lot alike, but we know Staph sapro definitely goes with UTIs, one way that labs can differentiate between them is using that novabiocin disc. So Staph epi, again, will be susceptible to that novabiocin. It will have that zone of susceptibility around it, whereas Staph sapro is novabiocin resistant. So that's one way. Staph intermedius, again, tends to look just like the others. I'm not going to do too much there. It is catalase positive. It is coagulase variable. There is a couple other staphs that might be coagulase variable from time to time, where they can be positive, depending on the strain. As a result, if you really wanted to start to separate, is it truly Staph aureus, which is a major pathogen, from the other staphs, you could do some extra testing. So if you did a VOPAS score, Staph aureus, remember, is P positive, and if you did a PYR, Staph aureus would be PYR negative. Whereas on Staph intermedius, it'd be the opposite. VOPAS score would be negative, PYR positive. So you can see how those additional tests can really separate between them. And the only thing that we really linked intermedius with is especially dog bite type wound infections. That's all you really need to take away there, and I think we're good. Micrococcus, lots of different morphologies for how it grows. It can be so many different colors, pink, white, tan, orange, okay. You get the idea, etc. Lots of different kind of looks to it. It is catalase positive, which is why it's grouped with the staphs, coagulase negative. This case, it would be oxidase positive if you did oxidase test it. We do know that oxidase is more of a gram-negative test to run, but just to keep that in mind if you're... And then as far as disease goes, it pretty much doesn't cause anything. It's contamination. It's a big normal flora of our skin. It's usually commonly found as contaminants. We mention it because you will see it pop up on a colony, on an agar plate in the lab at time to time. So you just should know about micrococcus, and it's pretty much always contamination. Very rare to cause disease. Okay, so we're good with staphs. Let's move on to our streps. Okay, so streps in general, again, are smaller colonies than the staphs, so they're a lot more tiny. And the biggest part with streps is their hemolysis. You have to know their hemolysis patterns, because that will tell you what types of tests you're going to run next. So remember, strep pyogenes is considered group A strep, and it is very much beta hemolytic. So again, we know it's a gram-positive cocci. We catalase test it, and streps are catalase negative. The next thing you do after you catalase negative, you should look at the hemolysis. Okay, I have a beta hemolytic. That will tell me to run my bacitracin A disc, and that should be susceptible. You could run a CAMP test. That would be negative. You could run a hip urate hydrolysis test, and that would be negative. You could run a PYR, and that would be positive. For disease, strep pyogenes, just so much, causes a lot. Skin infections, toxic shock syndrome, scarlet fever, let's see, necrotizing fasciitis. The list just goes on and on. Remember, there are those two, what we call post-streptococcal infections that can occur after you've had a strep pyogenes infection. And again, I've just got to write strep throat here. That's the main one. That is acute glomerulonephritis and rheumatic fever. Those are your two post-streptococcal infections that might, or complications that might occur. All right, strep Ehrlichiae, remember, is group B strep. And again, the hemolysis is beta hemolytic. So we see it's a catalase negative, it is a strep. We see it's beta hemolytic, so we're going to run those same tests: bacitracin resistant, CAMP positive, HIF urate positive, and PYR negative. So you can see how those tests will separate between our two main beta hemolytic streps. As far as disease, the only one I ask you to remember is neonatal meningitis. So I always say think group B for baby. So this is the one, again, that moms will get swabbed at some point in their pregnancy to see if they carry strep A galactiae in their vaginal tract so that they can be given antibiotics, get rid of it out of there before giving birth. All right, strep pneumoniae has no group name again. It is alpha hemolytic, so it's catalase negative. So once we see it's alpha hemolytic, you should be running your Optekin P disc, and it should be very much Optekin susceptible. You could also run a biosolubility test here, and that would be biosolubility positive. Disease, of course, it causes pneumonia, just like it's named for, which is the #1 cause of pneumonia, and it also causes meningitis. So two big diseases. And the other unique thing with strep pneumoniae to keep in mind is that it is a Gram-Pos diplococci. So it is paired up, and they are said to look like cat-eye shape or lancet shape, whatever one you want to call it. So kind of like that almondy look to it. But yeah, so Gram-Pos diplococci on strep pneumoniae. Viridin strep is also a group of alpha hemolytic streps. These are a lot more minor. They're all just kind of grouped together because there's a ton of other alpha hemolytic streps out there that just don't do a lot. Here and there they'll cause a disease, but they're not as major as the strep pneumoniae, so they just group them kind of together. Again, they're catalase negative. You would run your Optekin disc, and that would be resistant. You could run your biosolubility test, and that would be negative. So when they do cause disease, they tend to cause either like endocarditis or dental caries are typically the type of diseases they will cause. So common examples of this would be strep mutans, strep mitis are two common types of viridin strep. Enterococcus can be any hemolysis. The reason that we have Enterococcus with the streps is that it has more features to be like the strep than it does the staph, so we always put it here. This is the one that is catalase weak positive. It might be so weakly positive that you miss it and call it catalase negative, which that's fine. And sometimes you might hear people call this group D, but there are other group D streps out there, so just keep that in mind. But the test that we typically run on Enterococcus is going to be bioesculin, and it is positive in that. It also loves to grow in 6.5% NaCl broth. God, I cannot spell, and that's positive. If you did run a biosolubility, though, that would be negative. As far as diseases goes, it is connected with a lot of nosocomial type infections. So typically like a nosocomial-based UTI or something like that. All right, those are all our Gram-Pos cocci. Let's move on to gram-positive rods. So we start with our Bacillus species. These are Gram-Pos rods. Remember, these are very large boxcar-shaped Gram-Pos rods, and they form spores. So spore-forming large Gram-positive rods. So Bacillus anthracis is non-hemolytic, and this is the one that is said to look like Medusa head or comma projection. So they're very large colonies that usually have some sort of projection out of them. Lab test results go is catalase positive. It is still susceptible to penicillin. It is lecithinase positive. It is non-modal. That's all I really need you to know there. And of course, disease is anthrax, also known as that wool sorter's disease. Now again, there are three different three different types of anthrax infection depending on how they get it, whether it be through a skin infection, a lung, like an inhalation, which is more associated with bioterrorism, or a GI through ingestion. Bacillus cereus is beta hemolytic. These are also large colonies that are said to be just more spreading, so they don't really have the projections. Catalase pos, penicillin resistant, still lecithinase positive, and they are modal. So you have a couple of results there that will help separate them from the anthrax. Two diseases is all I asked you to know with this one. It's eye infections and food poisoning. Again, they form spores. Okay. Corynebacteria diphtheriae. Also, I don't know, for colony morphology, they tend to be gray, but if you were to grow them on that special cysteine tellurate agar, which is one of the test results, they would have that brown halo, which is very characteristic. So that could also be put under the test result here. Catalase pos. Let's see. There is other media that will grow Corynebacterium diphtheriae, which is Leffler's media, cysteine tellurite, and oh my gosh, I just blanked on the last one. Oh, it'll come to me in a minute. I'm going to skip it for now. There is one more. Go back and look in your chapter PowerPoint if I can't think of it here, but there is one more. Just totally blanked. I just had it. Of course, the disease that causes diphtheria, that is where it makes that classic membrane over the back of the throat, making it really difficult to breathe. Oh my gosh, terrible today. Back of throat. All right, whoops, go back. Okay. Listeria monocytogenes is beta hemolytic on the colony morphology. Of course, it's catalase positive as well. All of these in this little section were catalase positive. Hugely characteristic for its motility patterns. So this is the one that has that tumbling or the umbrella growth, depending on what type of motility test you ran. That is the biggest characteristic with it. It is also CAMP positive. And this, of course, causes neonatal meningitis. I think we've talked enough about this. You just had a question on it in your last lab report for week 10 as well. So, okay, I don't think I want to mention anything else with that one. I think you've had enough reference on that. It's really bothering me that I can't remember that last one. Oh, okay. Last of our Gram-positive rods, we have Erysipelotrix rhizopathia. I'm not going to worry too much about the colony morphology on that. As far as test results go, it's catalase negative. This is the only aerobic Gram-positive rod that is able to produce H2S. So that's a huge result for us there. And it causes exactly what it's named for, which is erysipeloids, basically skin ulcers, so severe skin infection. Typically getting this from handling animals. And so it'll pass through like an abrasion in your skin and then cause that skin ulcer. Nocardia are said to branch on their colonies. This is the one that is, I'm sorry, acid-fast, partially acid-fast positive. So unique in that, again, it's a skin infection is what it causes. Gardnerella vaginalis. The big thing here are the clue cells. So remember, those are squamous epithelial cells filled with the Gardnerella vaginalis bacteria. And so the disease it causes is bacterial vaginosis, so exactly what it's named for. This is the one that used to, I pray to God no one does this anymore. I think no one should do this anymore. Do that whiff test because it's said to have like a fishy odor to it. Oh, don't smell your samples, please God. Just don't. All right, and lactobacillus, the only thing we mention this for is that usually this is a really good bacteria to have. This is the one found in our yogurt, so it's very rare to cause disease. We want this. Typically they add it to the yogurts on purpose. It has good qualities for us. So I'm not going to say anything there. Okay, there was also some chapters on antibiotics. This was probably like week 3, week 4, I can't remember for sure. There was all those different antibiotics. There was examples of each category. The mode of action, remember, like does it, you know, inhibit the cell wall? Does it inhibit DNA synthesis? Does it inhibit RNA synthesis? So go back, revisit that. You guys have been tested on those a couple times. You should know what to expect for the testing questions on those. The other chapters that I didn't really put in here just because they were really recent that we just talked about them, and you've done a few study guides and lab reports really recently, so I'm hoping they're a little bit more with you, is the fastidious organisms. So remember, we did Haemophilus, Neisseria, Marxella, Francisella, Brucella, Legionella, which you had that whole discussion post on. So all of those kind of fastidious things, go back, review those chapters. And then our last, just week 10, we just covered the spirochetes with like syphilis, Borrelia, the intracellular organisms like chlamydia, some of the odd things like Ehrlichia, Cox, you know, Coxiella, things like that. They just did a study guide on, and the Mycoplasma. So those, we don't have that many questions that come out of the week 10 stuff. There's very minimal questions compared to the rest of everything that you had to know in Micro 1. So just do what you can. A lot of that should have been crossover from immunology if you've taken immunology or you're taking it right now. And so hopefully you guys will be fine. Just review your PowerPoints. I always say re-listen to any lectures of chapters that you feel weaker on. And especially, I think even more helpful would be to re-listen to the exam review sessions. We did 3 exam review sessions, so we've pretty much exam reviewed almost every chapter except for the week 10 stuff. And so you should be easily able to review that stuff again. I would go to those before anything else. Re-listen to those. You know, if you feel like you need to review an exam or quiz on how you did, sit down with your lab instructor or your coordinator at your location and see if they'll open it and allow you to quick look at some things if you had questions. So those are some ways to study. Now, as far as the test itself, let me give you some information. I just have to stop sharing my screen before I do this. Oh my gosh, yes, Gail. Okay, before I move on, Gail, thank you. That Crinibacterium diphtheriae, it's a brown halo on Tinsdale agar. And then cysteine tellurite and Leffler were the other 2 agars you can use. I had it just twisted in my head. So yeah, for Crinibacterium diphtheriae, one of the classic test results is that we'll give a brown halo in Tinsdale. And then the other 2 agars that used to be grown or sometimes still can be are Leffler's and cysteine tellurite. Thank you. I just had it a little bit backwards. I appreciate that, Gail. Okay. So as far as the test, let me get over to it. Oh, now I need to log back in. 1 second, and I can tell you how many questions and what type of questions to expect, because I think that's always helpful. So I'm just getting there. Hang in there with me. All right, so there's 119 points. 63 of those points are dedicated to multiple choice questions. There is 23 points dedicated to matching, 20 points dedicated to essay, and 13 points dedicated to short answer. So you truly have a variety of question types here. Now, when I say 23 points dedicated to matching, that doesn't mean there's like 23 matching. There's only 5 matching. But some of the matching obviously are worth more than others. But in general, 119, majority of the test is still going to be multiple choice-based questions, but there are some matching essay and short answer. And if you think back to your other tests that you've done and what questions were short answer and matching type, it's probably the same type of questions. I mean, I'm not that creative. It's the same kind of stuff. I'm a big believer in drilling it in over and over and over again. The more we keep hitting the same points, hopefully the more we remember them. That's my belief. So that's kind of how I test. I tend to ask the same things over and over to just try to get it in there. So hopefully that gives you a good idea of what to expect. If you have any other questions, just let me know. Everything needs to be done by this Friday. All of your tests, all of your lab practicals done by 5 o'clock this Friday. As far as your homework and anything else that's due, you know, there really isn't any discussion or anything this week besides tests. If you guys are missing something that you wanted to still try to submit, all course materials have to be done by midnight on Friday. But as far as the final exam and the lab practical, done by 5 o'clock this Friday. So make sure you get your appointments set up with your proctors or whoever it might be on your campus. If you have other questions, please feel free to reach out. Otherwise, you guys have a fabulous week. I've had a great quarter, and we will see all of each other, hopefully all of each other, next quarter in Micro 2 together. So that's it. Have a great night or day, whatever.